is controlled by three set of alleles, namely I, B, and I. The alleles I and IB are equally dominant, whereas allele I is recessive to the other alleles. Individuals with genotype A and I have blood group. Sons with genotype IB have blood group AB, while those with blood group O have genotype. Hence, if father has blood group A and mother has blood group B, the genotype of the parents will be father I and mother IB small I. The possible genotypes of other offspring would be IA, IB, okay, AB, IA, small I, and okay. Now we have another topic, that is pleiotropy. While a gene may have multiple alleles and thus give multiple genotypes, one gene may control several genotypes. For example, the recessive gene for white eye Drosophila, when present in the homozygous condition, affects several other features such as wing shape and shape of abdomen. Thus, a white-eyed Drosophila is also born with the vestigial wings and curled abdomen. Okay. Here, chromosomal theory of inheritance. It was observed by Sutton and Boveri in 1902 that maternal from mother and paternal from father, as we know, characters come together in the progeny, which is diploid or 2n and has chromosomes in pairs, and later on segregate during the formation of gametes. The gametes have a single chromosome from each pair and is haploid or n. Chromosomes from two parents come together in the same zygote as a result of the fusion of two gametes and again separate out during the formation of gametes. Chromosomes are filamentous bodies present in the nucleus and seen only during cell division. The above two observations proved that there is a remarkable similarity between behavior of characters during inheritance and that of chromosomes during meiosis. This led Sutton and Boveri to propose chromosomal theory of inheritance, and its salient features are as follows. Okay. The first one, diploid. The somatic body cells of an organism, which are derived by the repeated division of zygote, have two identical sets of chromosomes, that is, they are diploid. Out of these, one set of chromosomes is received from the mother, which is maternal chromosomes, and one set from the father is paternal chromosome. Two chromosomes of one type carrying same gene constitute a homologous pair. Humans having 23 pairs of chromosomes segregate. The chromosomes of homologous pairs separate during meiosis at the time of gamete formation. Third is linear location. The behavior of chromosomes during meiosis indicates that Mendelian factors or genes are located linearly on the chromosomes. With progress in molecular biology, it is known that a chromosome is made of a molecular DNA and segments of DNA are the genes. We have another question now. Who had proposed the chromosomal theory of inheritance? Sutton and Boveri proposed the chromosomal theory of inheritance in 1902. They linked the inheritance of traits to the chromosomes. Okay, we have another question that briefly mentioned the contribution of T.H. Morgan in genetics. Morgan's work is based on fruit flies, Drosophila, melanogaster. He formulated the chromosomal theory of linkage. He defined linkage as the coexistence of two or more genes in the same chromosome and performed a hybrid process in Drosophila to show that linked genes are inherited together and are located on X chromosome. His experiments also proved that tightly linked genes show very low recombination while loosely linked genes show very high recombination. Okay, now we have question number 11. This is done. 9 is done. A diploid organism is heterozygous for four loci. How many types of gametes can be produced? Locus is a fixed position on a chromosome which is occupied by a single or more genes. Heterozygous organisms contain different alleles for an allelic pair. Hence, a diploid organism which is heterozygous at four loci will have four different contrasting characters at four different loci. For example, if an organism is heterozygous at four loci with four characters, say A, capital A, small a, capital B, small b, dd, then during meiosis it will segregate to form eight separate gametes. If the genes are not linked, then the diploid organism will produce 16 different gametes. Okay. However, if the genes However, if the genes are linked, the gametes will reduce their number as genes might be linked and the linked genes will be inherited together during the process of meiosis. Two heterozygous parents are crossed if the two loci are linked. What would be the distribution of phenotypic features in F1 generation for a dihybrid cross? Okay. Linkage is defined as the coexistence of two or more genes in the same chromosome. If genes are situated on the same chromosome and lie close to each other, then they are inherited together and are said to be linked genes. For example, a cross between yellow body and white eyes and wild type parent in a Drosophila will produce wild type and yellow white progenies. It is because yellow bodied and white eyed genes are linked, therefore, they are inherited together in progenies. Elementary idea of polygenic inheritance. When a trait, feature, or character is controlled by a single gene, it's termed as monogenic inheritance. Many traits or features are controlled by a number of different genes. For example, the skin color of humans and kernel color of wheat. For example, the skin color of humans and the kernel color of wheat, okay, results from the combined effect of several genes, none of which are singly dominant. Polygenes affecting a particular trait are found on many chromosomes. Each of these genes has equal contribution and cumulative the total effect. Uh, 3 to 4 genes contribute towards formation of the pigment in the skin of humans. So, there is a continuous variation in skin color from a very fair to very dark. Such inheritance controlled by many genes is termed as quantitative inheritance or polygenic, poly meaning due to many genes inheritance. So table. So in polygenic inheritance, each dominant gene controls equally the intensity of the characters. The effect of dominant genes in cumulative. The intensity of character or trait depend upon the number of genes. Next topic is chromosomes and genes. Chromosomes. Eukaryotic chromosome. The label eukaryote is taken from the Greek for true nucleus and eukaryotes. All organisms except viruses, eubacteria, and archaebacteria are defined by the possession of a nucleus and other membrane-bound cell organelles. The nucleus of cell in our bodies contain approximately 1.8 meters of DNA in total, although each strand is less than 1 millionth of a centimeter thick. This DNA is tightly packed into structures called chromosomes, which consist of long chains of DNA and associated proteins. In eukaryotes, DNA molecules are tightly wound around proteins called histone proteins, which provide structural support and play a role in controlling the activities of the genes. Okay, a strand 152 200 nucleotides long is wrapped twice around a core of 8 histone proteins to form a structure called a nucleosome. The histone octamer at the center of the nucleosome is formed from two units each of histones H2A, H2B, H3, and H4. The chains of histones are coiled in turn to form a solenoid which is stabilized by the histone H1. Further coiling of the solenoids forms the structure of chromosome proper. Each chromosome has a P arm and Q arm. The P arm from the French word petit, okay, meaning small, okay, is the short arm, okay, is the short arm, and the Q arm, the next letter in the alphabet, is the long arm. In their replicated form, each chromosome consists two chromatids. The chromosomes and the DNA they contain are copied as part of the cell cycle and passed to daughter cells through the uh, process of mitosis and meiosis. Prokaryotic chromosomes. The prokaryotic Greek chromosome nuclei, okay, including Eubacteria and Archaebacteria, lack a discrete nucleus, and the chromosomes of prokaryotic cells are not enclosed by separate membrane. Most bacteria contain a single circular chromosome. There are exceptions. Some bacteria, for example, the genus Streptomyces, possess linear chromosomes, and Vibrio cholera, the causative agent of cholera, has two circular chromosomes. The chromosome, together with the ribosomes and proteins, are associated with gene expression. It is located in the region of the cell cytoplasm known as nucleoid. The genomes of prokaryotic are compact compared with those of eukaryotes as they lack introns, and the genes tend to be expressed in groups known as operons. The circular chromosome of the bacterium uh, Escherichia coli consists of a DNA molecule approximately 4.6 million nucleotides long. In addition to main chromosome, bacteria are also characterized by the presence of extrachromosomal genetic elements called plasmids. These relatively small circular DNA molecules usually contain genes that are not essential to growth or reproduction. They are used as vectors in genetic engineering. Genes. The gene is the basic physical and functional unit of heredity. It consists of specific sequence of nucleotides at a given position on a given chromosome. Codes for a specific protein. Genes consist of three types of nucleotide sequence. Coding regions called exons, which specify sequence of amino acids. Non-coding regions called introns, which do not specify amino acids. Regulatory sequences, which play a role in determining when and where the protein is made and how much is made. So for आज की वीडियो के लिए इतना ही. बाकी नेक्स्ट वीडियो में हम सर्च फॉर जेनेटिक मटेरियल एंड डीएनए एज जेनेटिक यहाँ से स्टार्ट करेंगे इस वाले टॉपिक से. तब तक के लिए लाइक शेयर